Hi everybody, this is just a quick tutorial to show you how to use BLAST to find information that you're going to use in your final project about a particular gene and the protein that it makes. So I decided for this example, I wanted to figure out what gene and protein is related to color blindness. So I'm just going to Google for color blindness gene and see what I can find. So we've got a number of different search results. I want to find something about the gene name. So I'll just try and find the first search result. Let's see what I found. It's saying that it's on the X chromosome, but it doesn't tell me the gene name on that web page, so I'll check something else. Oh, here's what is the gene for color blindness. Let's look. Still not finding it. Medline Plus will often have specific genes. Inheritance. Oh, this looks like what I want. Okay. So these look like gene names. So gene names usually are a few capital letters and some numbers sometimes. So it looks like there are two genes that are specifically related to color blindness. They're on the X chromosome. So I'm just going to pick one of those. Genes make proteins. And then I'm going to go to this NCBI website. This is the National Center for Biotechnology Information, and this is the really great place to look up genes. So instead of all databases, I'm going to search for a gene. I'm going to paste the name of that gene in and search, and we'll see what we find. All right, so it looks like this is the gene name, and it makes this protein called opsin-1 long-wave sensitive. It's the gene name in a human. And if we look down on these search results, we can see that in addition to humans, there's a similar gene for opsin-1 in chickens, it looks like, and cattle, and dogs, and dolphins. So a bunch of different types of organisms. So I'm going to pick just one of these that I'm particularly interested in. Maybe I'm interested in color blindness in humans. Maybe I want to research it in some other animal instead. So I'm just going to pick one. I'm going to go for elephants because that sounds really interesting. Of course, it means I'm going to have to go find information about color blindness in elephants after this. So I clicked on it. This is telling me a little bit of a summary about it, what the organism is, the genomic context. So it looks like this came from when it has an assembly and it looks like some big thing. That usually means that somebody sequenced the whole genome of this elephant. And what I'm really particularly interested in finding here is what we call the FASTA. So if you go down to where it says genomic regions, transcript, and products, go to the FASTA of whatever you looked for. So you're probably not going to be looking for color blindness gene. You're going to look for the whatever protein or gene you chose. So here it has the name of it, and then this FASTA is a particular format where it basically is showing you the list of all the A's, T's, G's, and C's in order for that particular gene. So it's pretty long here. Now you could copy paste this whole sequence into BLAST. But also, there's an option on the FASTA page where you can just go over to the right side under Analyze this sequence and just run BLAST straight from there. So BLAST is going to let us find similar sequences. So I'm going to run BLAST. It just put in the accession number, that's the identification number for that elephant color blindness gene. This sort of standard stuff is okay. You can optimize for highly similar sequences, that's okay. You might want to put, it's telling us it's going to come up with 100 search results. You might want to increase that to something like 1,000, especially if you're doing a human gene, because it might be that you just come up with all human results and you want to find some from multiple different animals. Otherwise, we can just click BLAST. And this is where it usually takes a little bit of time because it's trying to search for matches to that gene sequence or things that are close matches in this huge, huge database. That was not too long at all. So now I'm going to search down here, and it looks like, okay, the very top result is, it looks like it's coming from Loxodonta africana, which is our, I think it's an African elephant, if we could see that whole name. You could always look up, this is a scientific name, Loxodonta africana, but it's the scientific name of that elephant. It gives you these scores, which you don't need to worry about. The query cover, what that means is it means that 7% of what we searched for matched this thing that it found. So it's possible that we searched for something that's longer than this sequence that it found, but 7% of it matches up. And the part that matches up by 97% of the letters, the A's, T's, G's, and C's. So that's a really good match. So that's going to be very similar to, it's probably the same gene in that elephant with maybe just a few mutations in it. Maybe different elephants have slightly different sequence. The E-value, this you want it to be low. So if the E-value is really low, it means that it would be very unlikely for us to find a match for this in a random database full of random letters. So that's good. It means that we probably have a real match. We didn't just search for some short 5-letter thing that is going to be in practically every species genome. So you want low E values. All of these are really low. So when it has like 1 E to the negative 176, that means 1 times 10 to the negative 176th power. Anything to a negative number power is going to be like a super, super low number, like 0.0000 something. Anything that has an E and then a negative number afterwards is still going to be a super low number. So we have good E values here. They're nice and low. But you can see that some of them match better than others. So that first one matched what we searched for by 97% of the letters. This one matches by 92%, this one matches by 90%, etc. So now we want to just pick like 3 different species that we want to compare with each other. So unselect the select all. So I'm going to choose that first one, which is our elephant, and I'm going to look down and try and find some other kinds of animals or organisms that I want to compare this gene with. So I might have to like look up Canis familiaris. I happen to know that that is dogs. So this is probably the opsin gene for dogs. Propithecus coquerelli. I really don't know what that is. So I'm going to copy it and search for it and see what that organism is. That's the, it didn't. I'm going to copy the text, not the. This is a scientific name. It is Coquerel sifaka. It's a type of lemur, it looks like. That sounds like a fun one. So maybe I'm going to go for these lemurs. That one matches by 90% of the sequence. I'm going to go look down here. Well, it looks like we've got Hippopotamus amphibius. I think that one's actually a frog, even though it has hippopotamus in the name. Lutra, I'm pretty sure these are otters. So I just recognize some scientific names, but you're probably going to have to look up some of them. I have no idea what, like, this one is, Aeotis. I'm just going to copy that and search for it because I'm curious what animal this is. Oh, it looks like that's another night monkey from South America. It's a different continent. Well, it could be interesting to like compare from like different primates on different continents because the lemurs were in Madagascar. Maybe I'll do that. So maybe instead of the elephant, I'll find a different primate. I don't know what this is, so let's search for that and see if it looks like a primate. I don't want to copy the link, I just want to copy the text. Oops, search for too much, just that. Oh, it's a bat. Interesting. Okay, still looking for another primate, monkey or ape or something. Molossus molossus, that's an interesting scientific name. This is another bat. Huh. We have a number of bats in here. Desmodus rotundus, Cebus capucinus. This sounds like it could be a primate name, not sure for sure. That is a Panamanian white-faced capuchin. Okay, that's another primate. So we've got Central America, South America, and Africa. Maybe I will go with those three. So you can pick whichever 3 species you want, but know that you're going to have to research a little bit about this gene in those species afterwards. And you may be able to find lots of information. It may be just a little bit. After we've selected our 3, we're going to go look at the alignments. So it will take a little bit to load. The more sequences you have selected, the longer it will take to load. So I would recommend don't do more than 3 at a time. All right, so it finally loaded the results for me for the alignments. So I chose flat query anchored with dots for identity. So what this is showing, the query is the sequence that I originally searched for. And then these 3 are the accession numbers for each of those 3 organisms that I checked over here in the descriptions, which was this cat, a llama, and this, I think this one's a pig. And so you can see the sequence of A's, T's, G's, and C's on the query, which was the thing I originally searched for. And then on the 3 underneath it, anywhere there's a dot, it means it has the same DNA letter as the one in the query above it. Anytime there's something else, like for instance here, these 2 both have a T, whereas our query had a C. So in that case, this organism, the AP one, which I believe was this was the cat. So the cat has the same sequence in that point as our original query, whereas both the llama and the pig have a T there instead of a C. In some areas, you're going to see what looks like a blank sign or a dash. In that case, it means there's been a deletion where that organism is missing a letter at that point in their DNA. So on this one, both the query and the cat have an extra letter as compared to the llama and the pig for this gene. So when you are doing your project, you're going to need to include a screenshot of at least 3 rows of your alignment here. And so try to pick one where there's a few differences, where you definitely have all 3 of the things that you checked are in there. So you could pick something like this. To screenshot, you would hold down the Control key and press Print Screen. Some people have an add-on that lets you screenshot something else as well, so I'm just going to screenshot. And that basically will copy this portion of the screen, and then you can go into something like Microsoft Word, and you can just paste it, and there's your screenshot. So if you're doing this as a paper in Word or something like Google Docs, you can just paste it in there. If you're doing it as a PowerPoint presentation, you can paste it in there. Let me open up PowerPoint so that you can see. If you are doing your project as a presentation, I'm just going to open a new presentation. You can choose something like design to pick a PowerPoint design for yourself. You can change the design later if you want. You can write titles on here. Whatever your title you want to be. And then if you go up to the Home tab, you can add new slides. Another option that you have is you can go to Slideshow and record slideshow. And then there should be a record button that you can press so that you can talk and it will record the audio. And then if you turn it in like that, you don't have to have every single thing written on your slides. You can just have a few bullet points, main point, another main point, etc. And then maybe a picture, and then you can talk about it and record that talking in the background so that it's not quite so word heavy. You get a little more variety that way. You're also welcome to do this as a paper. You could do it as a video. If you are into making websites or something, you could even make a website. I'm not too picky about your format as long as you have all the information in there. And make sure that you take a look at the project instructions so that you know what to include. So this is your project instructions. It's in Canvas. It includes details about how I'm going to grade each part and how much points it's worth. So just make sure that you hit all the bullet points and have all the information there. The part about BLAST is right down here. So this is what you need for the BLAST results. And hopefully that helps.